right, hi everybody. Thanks for coming to the CPEP Seminar. It's got 1 p.m., so we're going to get started. Today our speaker is Max Beal. Max is a PhD candidate in Civil and Environmental Engineering, advised by Paul Block in the Water Systems and Society Lab here at UW-Madison. His research focuses on hydrology, climate variability, and impacts on water quality with a focus on forecasting. Max helps to develop and distribute season-ahead forecasting of harmful algae to improve beach and lake management for Lake Mendota, a project with Water Systems and Society Lab that they've overseen it since 2015. So I'll pass it over to Max.
Cool. Thank you very much for the introduction. I'm going to see if I can turn this on. Hopefully that'll help. All right, yeah, well, thanks so much. Thanks so much to CPEP for having me, and thank you all for coming out. I work with Dr. Paul Block and I'm going to be presenting today on some of my dissertation research, which here I've titled Hydroclimatic Forecasting and Satellite Remote Sensing for Harmful Algae in Small Inland Lakes. So before I kind of get into the meat of the presentation here, I wanted to start out with kind of an overview and a little bit of a plug for our lab and what we do. So I'm in the Water Systems and Society Lab, and I generally think of the research that our group does as having kind of 3 main components. The first of those is understanding environmental processes. So for us, that often means understanding the seasonality of some variable of interest. So in this case, with harmful algae, we're thinking a lot about when is it most productive, when is it most abundant in our lakes, which you all might be able to guess is often the summer. And then also understanding hydroclimatic variability. So variables like temperature, precipitation, how they vary across the year, between years, and how the magnitude of that variability influences an outcome of interest. Finally, understanding those different things at different temporal and spatial scales. As was mentioned, I'll be talking a lot about season-ahead forecasting today. So we then take that kind of understanding of environmental processes and often apply it to modeling systems of interest for response and prediction. And so we kind of span a broad variety of topics in water systems and society, but they're all pretty much water related. Today I'll be talking about water quality extremes. We often do work on water quantity extremes and things like water related disease, etc. And then finally, taking these kind of first 2 components, and as we're doing this analytical work, really thinking about how these things fit into informing decision making. And so for us, that often means building things like forecasting systems, monitoring tools, which I'll talk about today, and then also coming up with systems for forecast-based action. So how can our forecasts be implemented into decision-making processes that kind of help us manage different systems. So I won't talk about all of these things here today, but I think I'll cover a good portion of them. And so with that, I'll go ahead and just introduce the 2 parts of my talk today. So I'm going to start in part 1 talking about season-ahead forecasting of peak season algae metrics in the Midwest and Northeast US. So I'll start by talking about a forecasting system that we've developed for Lake Mendota, specifically for cyanobacteria biomass. And then I'll talk about some work that we've done to broaden that approach out to look at lakes all over the Midwest and Northeast US. And then in part 2, we'll come back to Lake Mendota, focus in on the development of a satellite remote sensing monitoring system for harmful algae indicators using the Sentinel-2 and Sentinel-3 satellites, if you're familiar. So with that, I'm going to go ahead and get into this very first part. So a season-ahead forecasting system for cyanobacteria and beach closings on Lake Mendota. And so this work was done in conjunction with a bunch of other graduate students, Brian, Caitlin, and of course, Professor Block. So I'll start out by giving you a little bit of motivation. I think a lot of you in this room, having lived in Madison, might know that we have a problem with harmful algae blooms, particularly in Lake Mendota. And so we've actually had problems with algae blooms for a long time, believe it or not. There's records of algae blooms all the way back in the 1800s, and then they really became a sort of chronic problem in the early 1900s, fueled by municipal wastewater discharge. And more recently, of course, agricultural runoff has really maintained this high state of productivity in the lakes with lots of nutrient runoff into the lakes. that allows these cyanobacteria to really grow and in some cases create a bloom, which creates these scums on the water's surface. And the real threat with cyanobacteria blooms is that cyanobacteria are potentially toxin-producing organisms. So when you get a bloom, when you get a lot of biomass for cyanobacteria, there's a greater chance that you could get toxin production, and that of course has negative effects for human and animal health, but also for ecosystem health, and as a chronic issue can have effects on our economic opportunity around these lakes. This, of course, is not just a problem for Madison. It's a problem much more broadly. We've seen problems with algae blooms in our Great Lakes, of course, Lake Erie, and then also Lake Superior in recent years. And then there's been some work done to suggest that in some lakes, climate change might fuel more cyanobacteria blooms going into the future. I'll also just point out that there are tons of small inland water bodies across the Northeast and Midwest US that are in kind of this high risk zone for water quality impairment. And that's just because we live in a very agricultural and kind of wet region in the Northeast and Midwest US. And so there's a lot of lakes here that could potentially benefit from management tools to try to help people who live on these lakes manage this harmful algae problem. So the tool that I'm going to be talking about today are seasonal or season ahead forecasts. And to kind of explain that, I have this diagram here. And if you can imagine for me that in the blue area of this screen, it's still springtime, and as we move across the screen towards the lake, we get into summertime. We know that variables in the spring, like frequent and intense precipitation events, warm temperatures, and high phosphorus loads, combined with early summer variables like available sunlight, grazing by zooplankton, and stratification strength of our lake, can all combine to influence cyanobacteria productivity and abundance. And so our question here is, can we use these local scale variables that I've just shown you, and then also global scale climatic variables that might modulate what's going on near Madison to forecast cyanobacteria biomass ahead of the summer season? And so the reason we might want to do this is because this provides water quality information ahead of what I'll call the high risk season, right? Summer is when cyanobacteria is most abundant, but it's also when we're using our lakes the most. And so there's this period where we are likely to come into contact with it. Season ahead forecasts can work a little bit differently from shorter term forecasts because they provide information at a time scale where you can maybe make different decisions. So that might be something like ahead of summer, you can maybe still change your overall budgeting for the summer. You can maybe change testing routines on beaches. And you might be able to hire and train more lifeguards, things like this. And lastly then, of course, to increase public awareness about what the season might look like. So a little bit more technically, how we do this is borrowing this kind of time scale again. We would start in the spring by identifying some preseason predictors, so some of those things that I was just talking about. So here we've identified the preseason as spring, or March through May. And so we pick out some of those predictors, we do correlation analysis, see which ones might influence cyanobacteria abundance. We put those things into a principal component regression model, and then we come up with a forecast. And so this is actually the forecast from 2023. We forecast average cyanobacteria biomass for June, July, and August, so all in one, that whole season. But these are ensemble or probabilistic forecasts. So that just means that we come up with a bunch of different potential scenarios, and then we bin those scenarios into categories. So the categories you see here are below normal, near normal, and above normal. That's just based on the long-term record for cyanobacteria in Lake Mendota. So below being the lowest third that we've seen in the lake across this time series, near being the middle third, and above being the upper third. And this kind of helps to convey, ideally, to managers and other folks who might use this forecast, that the sort of inherent uncertainty in our models. So here we forecast for this year a 50% probability of near normal algae, so about average, and we release that on June 1st. And then we actually update this forecast. This is where the forecasting system kind of comes into play. So we pick out new predictors at the beginning of the summer, so these within season predictors when conditions might have developed a little bit more. We create another model, and then we're able to release a new forecast. So I don't know if those of you in the back of the room can see this, but with this subseasonal forecast that's released on July 1st, we actually saw a big shift from near normal conditions to below normal conditions. And while we don't really have the data to verify this yet, from some work from our monitors with the Clean Lakes Alliance, we've seen that it's a pretty good water quality year in Lake Mendota. So hopefully that actually plays out in the cyanobacteria biomass data. So a big part of building these models is going through a validation process to make sure that these models actually work or have some skill, we'd say, before we try to forecast anything and release anything to the public. And so I'm going to talk a little bit about how we validate those models. First, I want to just talk about some of the predictors that we ended up using for the season ahead forecast. So to orient you, on the upper right-hand corner here, we have this map that shows some different locations where we got our data from, precipitation data and stream flow and things like that. And so some of the predictors that we use for our season ahead forecast are spring, MAM stands for March, April, May, extreme precipitation events, April total precipitation, average stream flow from the Yahara into Lake Mendota, and then also things like external phosphorus loads. We also then use equatorial Pacific sea surface temperatures, and this gets at these teleconnections between global climate phenomena. You can think of that as anomalous heating and cooling of the oceans that drive precipitation and temperature patterns largely over the US and the Midwest. And so these are very slowly developing processes, and so we can pull on them at a season ahead time scale to predict what conditions might look like near Madison. So below this, I have what we call a hindcast. And so this is where we take our entire time series and we validate how well our model works before we release anything. So we do what's called leave one out cross validation. We pretend we can't see 1 year, and then we build our model and we try to predict it. And so these whisker plots here show the ensemble of predictions that we have for each year. So that's the range. And then the red line shows what actually happened. So that's actual average cyanobacteria biomass. And hopefully what you can see is that we generally are able to get this red line within our boxes. And so we're at least capturing some of the variability in cyanobacteria biomass with this forecast. You can also look at forecast skill categorically. So by comparing the category that you forecasted versus the observed one. And so on the diagonal here, we have the number of forecasts that we got right for each category. So when we observed a below normal year, did we forecast a below normal year? And generally, we see this forecast does a pretty good job. We do have a few misses. Sometimes we'll forecast a below normal year, but it's really a near normal year. But we don't have any extreme misses. And so that might be a scenario where we say it's going to be a below normal year, but it actually is like a really high cyanobacteria year. And so that's encouraging for us from a management standpoint. So I won't talk about this for too long, but we also build these forecasts for beach closings. So right here, this is a graph of days closed on 3 beaches in Madison. You can see that there's quite a bit of variability in how many days a beach is closed due to cyanobacteria. And so we go through a very similar process to build a forecast for these beach closings. And they tend to do pretty well. There's some do better than others. But really what I want to point out here is that if you're interested in receiving these forecasts, the beach days closed and the Mendota cyanobacteria forecast, feel free to email myself or Dr. Block, or just come up to me after the talk and we can get you on a mailing list, and that'll come out again on June 1st of next year. So that's kind of the end to this part 1 of part 1. And now I want to talk about how we've taken this approach that we developed for Lake Mendota and expanded it to many lakes across the Northeast and Midwest US. Developing this 1 season ahead forecast got us thinking, can we apply this same approach to a variety of lakes using some list of pretty readily available predictors, and is there any skill, is there any value in doing that? So again, I will just reinforce that there are a lot of small water bodies across this region of the Northeast and Midwest US. And so we're pulling on this data set, the Lagos US data set, which finds lake data for all of these small inland lakes to build some of these models. And so there's many, many water bodies that could potentially benefit from having some sort of management tool. So we went ahead with this Lagos data set and identified 178 lakes with at least 15 years of chlorophyll A data and a sampling frequency of roughly 14 days throughout the summer and early fall. This is all following a lot of work and collaboration with Professor Wilkinson over in the Center for Limnology. We're using chlorophyll A data here as our value of interest because it's a lot more readily available than something like cyanobacteria biomass. And then we came up with 3 metrics. Magnitude, or the mean seasonal chlorophyll A concentration, severity, which is the 95th percentile of historical chlorophyll A concentrations. So you can think about that as another category. And then duration, or the percent of sampling days that chlorophyll A exceeded some regionally defined value that we pulled from the literature. And so I'll talk briefly again, technically, about how we built these models, but it's a very similar approach to what we've done for Lake Mendota. So we start out by, in the springtime, identifying some of those preseason predictors for each lake. We create a statistical principal component regression model, and then we perform a model validation, which is a leave one out cross validation. We then repeat that process for all these different metrics. And the reason that we might want to do this for these 3 different metrics is that ideally different metrics can answer different questions for managers. So something like, will algae be a problem this season in my lake at all? How long is it going to be a problem for? Is there going to be a long duration of high algal biomass? And what are the chances of those really severe events happening? Something like a toxin production event or a fish kill. So after doing all of this analysis, we would again release that forecast for the metrics at the beginning of our season of interest, which here we've defined as summer and fall. And then again, that forecast might look something like this. So here, if we are trying to forecast, we have some metric of algae abundance on the y-axis, and if we're interested in forecasting the very last year, our forecast would look like this green area, where we come up with a bunch of different ensemble members, and then again, bin them into these different categories, and those are based on the historical observations for each lake. So we have a low, normal, high, and then severe category. So we needed to start by taking these 178 lakes and looking at the chlorophyll A time series in each of them. And so what we've done here is taken those lakes and we've separated them into Midwestern lakes and Northeastern lakes. You can see that there's some interesting differences in average chlorophyll A between those 2 lakes. So the Midwest generally has higher chlorophyll A than the Northeast, and so we've split these lakes for that reason and a few others, which I'll get into later. So by looking at this graph here, you can kind of pick out that there are 4 months in which chlorophyll A peaks for both lakes. And it's what you might expect the middle of the summer through early fall, so July through October. And this is what we've identified as our peak season. And so for the season, we're really trying to predict 2 numbers. The mean log chlorophyll A and the proportion of elevated chlorophyll A samples. And again, that's just using a previously defined threshold that we pulled from some papers. That means that the previous 4 months, March through June, are going to be our preseason. And so we're looking at predictors from this preseason of March through June at a few different spatial scales that can ideally get at some different processes that are going on. So at the within lake scale, we use preseason chlorophyll A. At the watershed scale, we're looking at things like total precipitation, extreme precipitation events, mean temperature, and soil moisture. And then at the global scale, we're looking again at these regions of sea surface temperature that really get at these global teleconnections. So we identify these predictors from these readily available data sets for each lake, and then we test correlations between those predictors and our outcomes of interest at the 95% confidence level. So before I kind of get into the results, I want to talk for a second about how we identified relevant areas of sea surface temperature. So you can see here that we've split up the area of the country into the Northeast and the Midwest here. And our idea here is that we would identify different regions of the ocean that might influence the Midwest versus the Northeast. These are both large regions. They're both close to different oceans, and so they might have different influences, or might be influenced by different regions or oceans. So the way that we've done this is we would start by taking one chlorophyll A time series from a lake in the Northeast. So this is some lake and it's chlorophyll A, and then we'd correlate that time series with every available sea surface temperature grid across the world, and then we'd pick out the statistically significant correlations. So we then repeat that process for every lake in the Northeast, picking out these statistically significant regions, and we add them up. And that gets us a map like this, where we can see the number of significant correlations. And it starts to get at the idea of there are certain grids in the ocean that appear to have a consistent influence on the lakes in these regions. So we've done this for the Northeast and the Midwest, and then we pick a threshold, 20 lakes in the Northeast, 10 lakes in the Midwest, and that's how we identify those regions. We then take principal components of those regions to try to get the dominant signals coming from each of those sets of grids. So with all of that sort of predictor identification out of the way, we can do all this correlation analysis and get the results, and we find a few interesting things. So for our magnitude metric, we find that 135 lakes out of our 178 candidate lakes had at least one significant predictor. And so here you can see in these graphs on the upper right that preseason chlorophyll A was far and away the most important predictor of magnitude across these lakes, followed closely by the first principal component of sea surface temperature, and then extreme precipitation events. So we're seeing some representation from each of those kind of spatial scales. Duration's a little bit different here, so we see only 82 lakes had at least one significant predictor. We generally see the same predictors float to the top, but more sea surface temperature representation, and then preseason chlorophyll A and air temperature also seems to play an important role. Generally though, seeing that the watershed scale predictors are falling out a little bit more, and that might be because we're using gridded data sets, so not gauged data sets. And it also might be because there's kind of a lot of variability going on between this spring season and the summer season, so it's kind of a long time frame. These maps over here just show in which lakes had certain predictors. So which lakes had sea surface temperature, which ones had preseason chlorophyll A as a significant predictor, which had both. All I really want you to take away from these are that there's a lot of different colors mashed together. We originally had thought maybe there will be sort of regions of the country that lakes would have similar predictors, but we're really not seeing that. And so that kind of raised this question to us of, are there other variables that might be influencing which lakes retain which predictors and forecast scale more broadly. So before I talk more about that though, we also had to do a hindcast analysis. So we came up with our predictors, we went away and built our models, and then we assessed how well these models did. And so that's represented here by these two graphs. And so I'll walk through these slowly here. So over here on the lower left-hand side, this is our magnitude graph. Along the x-axis, we have the observed category. So that's what actually happened in terms of algae magnitude. Along the y-axis, we have proportion. And then each bar here is colored by the proportion that we predicted. So this gray box here represents the low observations that we predicted would be low. So those are correct forecasts, and kind of so on and so forth. I'll argue up here that lake managers are likely most interested in accurate forecasts of high algae abundance and duration, because that's when it's usually a problem. And so overall here, in summary, we found that 50% of those elevated magnitudes, so the high and severe category, were accurately forecast. And we found that 67% of our high durations here were also accurately forecast. And so that means that we're beating what we call climatology, or just kind of averages. You can kind of imagine that for duration, if we have two categories, you have a 50-50% chance of getting it right. And so seeing that we are above that 50 means that these forecasts are adding some skill. We can also look at this on a more lake-by-lake basis. So here we've just come up with some maps where these closed circles represent lake models that predict those high and severe events correctly more than half the time. And so we found that 55% of magnitude models can do this, and 86% of duration models can accurately forecast those high durations more than half the time. I'll again just point out here that there's quite a bit of variability spatially, where forecasts do well and where they don't do quite as well. And so we're interested again then in some other variables that might be influencing forecast skill among these lakes. So I'll just point out or talk about two variables that we've looked at here. So lake area and the trophic status index. And so here on the y-axis we have the percent of high and severe categories that were forecast correctly. And on the x-axis we have different categories of lake area and different trophic status indices, where eutrophic lakes like Lake Mendota are most likely to produce algae blooms, have a lot going on, and oligotrophic are very nutrient poor. So generally we don't see a ton of differences between these lakes, but we found that the smallest lakes and the most eutrophic lakes we had the most difficulty predicting most of the time. We also found, though, I'll point out that a lot of very similar lakes could have very different skill scores. So these are two lakes in New York. They're about a mile apart, Maryville Lake and the Duanesburg Reservoir. They're both categorized as eutrophic, and they're roughly the same size. Maryville Lake, though, had a very high R-squared score here. It does very well, it predicts all those high events, and the Duanesburg Reservoir does very poorly. They have a different set of predictors. So Maryville Lake retains both sea surface temperature anomalies and pre-season chlorophyll A, while this reservoir just retains sea surface temperature anomalies. And I think this gets at some limitations to this modeling approach. So while we found that global climatic influences can do a pretty good job at indicating how conditions are going to set up for the summer around these lakes, they obviously can't get at what's going on inside the lake. And for a lot of our lakes, at the same time, pre-season chlorophyll A does a really good job of indicating what's going on inside the lake in the lead up to summer. But it can't do that for all lakes, right? There could be different food web interactions and other biochemical processes that we just aren't capturing here at all with this one predictor. And so that might explain some of these differences in skill. So in conclusion, we found that global sea surface temperature anomalies and pre-season chlorophyll A really stand out as the most common sources of predictive power across these lake models. We found that models demonstrated skill in the prediction of magnitude and duration of peak algae, and that forecasts of high and severe algae magnitude tended to perform best in large mesotrophic and oligotrophic lakes. So I'll also plug that we published on this somewhat recently, and so if you're interested in reading more about this work, you can talk to me or go ahead and find it online. So that kind of brings us to the second part of this talk, which will be a bit shorter. But we're going to zoom back in here to Lake Mendota and on a much finer time scale of really days to weeks, and talk about the development of satellite remote sensing system for harmful algae indicators for Lake Mendota. And these are just images from these two satellites that we picked out here. And this work was done with Professor Ozdogan over in Forestry and Wildlife Ecology. So to go back to the motivation a little bit, hopefully I've convinced you, or didn't need to convince you in the first place, that harmful algae is somewhat of a problem on Lake Mendota and a problem across the country. And I think that's well illustrated by this map in the middle of our slide here. And these are news reports of algae blooms from 2010 to present. I think this is a couple years old at this point. But I think what this really indicates is that people are seeing algae blooms on their lakes and are getting concerned about it, and they're reporting it. So while this is a pretty widespread problem, I think there's a dot maybe in almost every state, I'll also argue that it's a little bit of a difficult to define problem. And that's because harmful algae blooms can really range in their impacts. So they can have, aesthetic issues where people just don't like to see them. They can produce taste and odor compounds that make water not very nice to drink. But then they can also produce toxins, which can have really serious impacts on health. And so when we see an algae bloom like this one, our question is often, is this algae dangerous? You might be able to hazard a pretty good guess for this bloom in particular, but you'll just have to trust me that it's not always this cut and dry. So the way that we typically go out and figure this out is by taking a water sample, putting it under a microscope, and then identifying all of the different algae species in that sample. So if that sounds like it takes a lot of technical expertise and time, it does. It works well, but it also means that we're motivated to find ways to supplement that kind of sampling. And so we'd like to be able to use satellite remote sensing and to capture these trends across a lake spatially and at a faster time frame. And so we can exploit the dominant pigments in these two groups of algae here. Today we'll just talk about green algae, which are non-toxin producing, and cyanobacteria, which are. So chlorophyll A for green algae and phycocyanin for cyanobacteria, and try to use satellite remote sensing to characterize blooms using these two pigments. So the way that we'll do this is by taking two satellite missions, Sentinel-2 and Sentinel-3, and then developing some machine learning models to try to retrieve these three different metrics. Chlorophyll A to get at green algae, phycocyanin to get at cyanobacteria, and then the phycocyanin to chlorophyll A ratio. And that kind of gets at, can we characterize a bloom? Is it dominated by one or the other? We'll then compare some of those metrics to our water quality sampling data, and then do some post-processing to try to get at dissolved oxygen, and I'll talk about how well these models work across space. So, to give you an idea of why we picked these two satellites, they have some interesting trade-offs between them. So in the upper right-hand corner is an image from the Sentinel-3 mission, the Ocean and Land Cover Instrument. So it has a pixel size of roughly 300 meters on our lake. So that's pretty large, but it has a 2-day revisit time. So it comes over our lake quite often, and so we can get a lot of images of our lake. Sentinel-2, comparatively, has a 10 to 60 meter resolution, so really fine pixel size. We can get really up close to the lake, but I should say it has an 8-day revisit time, so we're only getting that image roughly once a week. If I can direct your attention over here, this black line shows an example spectral emission response for chlorophyll A, and what that just means is that when you look across the electromagnetic spectrum, this is where you tend to see chlorophyll A light up, so that's the kind of signal that we're looking for. And then these bars behind this black line represent where Sentinel-3 and Sentinel-2 can pick up spectral information. So you see a lot more blue here, which corresponds to Sentinel-3, and so that means that Sentinel-3 has what we call a better spectral resolution. So it can pick up information in more places of the light spectrum than Sentinel-2. So once we picked those two satellites, we of course had to do some water quality sampling. And so we've gone out on Lake Mendota since 2019, roughly once a week, and we've sampled at 35 different locations across the lake that are randomized in each of these grid boxes here. And the reason that we've done that is to try to get a really good spatial representation of the lake. And so you'll see us use these grid boxes again later in the talk. And so we perform this sampling to get chlorophyll A and phycocyanin using a YSI exosonde, and we do this when the satellites are passing over, so we can compare our observed data to our satellite-based estimates. And so once we have that data, we can start by looking at our image of our lake, and we can extract that reflectance information at the points where we sampled. We can then plot that spectral information against some of our harmful algae metrics, and so that might look something like this, where we have here phycocyanin on the y-axis and band reflectance on the x-axis, and so this is colored by each band. So those are those locations where the satellites can pick up information. You can do this again for chlorophyll A and for phycocyanin to chlorophyll A ratio, and really all I want to point out here is that to my eye there's not a lot of really clean linear relationships to pick out from these graphs. And so when we're trying to solve this problem of turning reflectance data into water quality, many people have chosen to use nonlinear modeling methods or machine learning methods to try to do this. And so we've picked two machine learning methods, random forest and artificial neural network, to try to do this. And I'm just going to dive into some of the modeling and validation of those models right now, starting with Sentinel-2. So in this slide, in the upper right-hand corner, we have plots of observed chlorophyll A on the x-axis to modeled chlorophyll A on the y-axis, so based on satellite data. And essentially what we want here is for those green dots to get as close to the red dotted line as possible. We also have some skill scores down here for how well these performs. So for random forest, we have an R squared of roughly 0.47, an artificial neural network 0.44. So roughly in the same skill range, seeing a decent R squared for this result. For phycocyanin, we see slightly better skill scores, again similar for the two models. And for the phycocyanin to chlorophyll A ratio, we're again seeing pretty good skill. Some scatter, particularly in the higher values, but generally the story here is that Sentinel-2 is doing a pretty good job of modeling these harmful algae indicators. Sentinel-3 is a little bit of a different story. So we have, for chlorophyll A, again, actually decent skill, again in the 0.4 range, but maybe with a little bit of bias in the artificial neural network in those upper ranges. For phycocyanin, we really see a breakdown of skill, so we're not really getting anything from these modeled results. And sort of similar for the phycocyanin to chlorophyll A ratio, where we actually get a decent R squared, but I think it's likely driven by performance at these really low range of values, and then you see a lot of scatter when you get kind of above 1. And so what we think is going on here is that there's just a mismatch between the spatial resolution of Sentinel-3, those 300 meter pixels, and where we're sampling, which is a really small point measurement. And so we think that mismatch might be causing some of this lower skill here. With that being said, there is still some skill to these models, and of course to the Sentinel-2, and so our thought next was how can we get at some other water quality monitoring variables that we might be interested in this case dissolved oxygen. So dissolved oxygen is what you'd call non-optical, so it doesn't change the color of the water, but it's an important variable for monitoring lake health because it's really important to aquatic organisms. And it's also can be closely related with algae productivity. When algae are photosynthesizing, dissolved oxygen goes up, and when they die off, they can use up a lot of dissolved oxygen. And so our thought here was, can we use these measures of algae to try to predict dissolved oxygen concentrations in the water column? So here we find that when we use these three variables in a random forest model, we're actually able to approximate dissolved oxygen pretty well. This is all in situ data, so this is data that we've observed, nothing to do with satellites here. When we then replace this in situ data, though, with our remotely sensed estimates, we actually find that we're able to predict dissolved oxygen using Sentinel-2 and Sentinel-3 quite well. Sentinel-2 much better than Sentinel-3, but roughly in the range of skill that we have for our other models. So finally, with these kind of 4 water quality variables that we can kind of get at through these satellites, we wanted to see how well these models perform spatially, right? That's an important part. Water quality varies a lot across a lake, and we want to see if these models can capture it. So here we've just aggregated mean absolute error, so that's just a metric comparing our observed data to our predicted data up to these grid boxes. So just remember those grid boxes were regions that we were randomizing points for sampling in. So you can see that there's quite a bit of variability in error across the lake, but also between metrics. And that kind of gets at this idea that each of these models has a pretty unique job in trying to pick up variability across the lake. The general story for Sentinel-2 is that we see pretty low error across the lakes. There are a couple problem spots. For chlorophyll A, you can see that kind of the inlet from the Yahara into Lake Mendota has a high error, and that might be because there's a lot more going on there. We also see some spots near picnic points. There might be some land interference there, things like that. Sentinel-3, we actually see pretty good performance for chlorophyll A, but then we see a lot more hot spots for our phycocyanin to chlorophyll A ratio and dissolved oxygen. So we're just generally seeing higher error across the lake. So in conclusion, for this part of the talk, we really found that Sentinel-2 and Sentinel-3 did both show skill for algae metric retrieval on Lake Mendota. Sentinel-2 generally outperforming Sentinel-3. We found that models based on in situ data allowed for indirect satellite-based estimates of dissolved oxygen, so able to approximate that non-optical variable. And then finally, we found that Sentinel-2 was able to effectively capture the spatial variability in these water quality metrics across the lake. So that is all I have for you today. I'd like to thank my research group and our funders, and of course the NTL LTR for making a lot of this work possible. And I'd be happy to take any questions.
Questions for Max?
Please remind me the difference in the two satellites. Number 2 was more frequent coverage.
Yeah, so Sentinel-3 is more frequent coverage.
Oh, it's more frequent.
Sentinel-3 is more frequent, but then that spatial resolution's not quite as good. So Sentinel-2 comes by roughly once a week, and that's the one that we have pretty good estimates for.
Okay, I'm still confused. Which one is?
I can maybe try to go back to these.
Sorry, I'm being
Oh, no, totally fine. So Sentinel-3, that image on the top right, it's kind of hard to see the pixels in the image here, but so that one has larger image pixels, which means that we're not kind of getting as good of a spatial image of the lake, but it comes by really frequently, and those things often are kind of traded off because of where the satellites was.
Okay. And I had another question, which is, so you said that you divide the Northeast and the Midwest. Do they have different areas of the world's oceans that they're teleconnections to, and what are they?
Yeah,
Where are they?
we generally see, see if I can go back and find this image again. So these, you can kind of think of these maps. So down here's the Midwest, up here's the Northeast, and those highlighted areas are generally regions that are more important. And so for the Northeast, you generally expect some regions in the North Atlantic Ocean to have a little bit more influence. For the Midwest, we generally expect that there's kind of less influence from oceans overall because it's right in the middle of the country. So that's kind of generally the pattern that we see here, but still the Midwest receiving some influence from the North Atlantic Ocean is our hypothesis anyways.
I was a little surprised that the ocean component seemed to be more influencing the results than the precipitation. Because my intuition tells me if there's not a lot of precipitation, there's not a lot of runoff, which I think was something that happened this year. We had a very dry May and June, not much algal blooms in the lake. So I'm kind of curious if the precipitation extremes and the amount of precipitation is somehow cross-correlated with what you're seeing in the ocean temperatures, and that's how do you account for that possibility?
Sure. So I think there's a couple things. So a big part of this is that we're looking a season ahead. And so for Lake Mendota in particular, I think that springtime rain and stream flow does seem to be pretty important in setting up conditions for the summer. But generally, something like sea surface temperatures in other sort of seasonal forecasting tends to work a little better at that time scale because you're still kind of representing the same things like temperature and precipitation. But they develop a little bit more slowly. So when we see that signal in the springtime, we can often be a little bit more confident that it's going to carry through to something like the summer. Whereas for Lake Mendota, for example, right, we did have that really huge bloom in the spring, right, and that coincided, I believe, with a very wet spring at the same time, and then things really dried up. So there's some of those within season dynamics going on too that can, I think, really change things with a variable like precipitation, which can vary a lot more in kind of its magnitude than sea surface Does that kind of answer your question?
Yeah, it's a nice presentation, Max. I'm really interested in the first part about the forecast for Mendota. And am I remembering right that you didn't have a significant predictor for spring temperature? Was it all precipitation based if it was meteorological?
Yeah, and that's actually something that kind of popped out in a sort of larger scale version of this too, which, sorry, did I cut off your question
No.
here? Okay. So I'll also say we're using air temperature here rather than water temperature, and I think that's just another one of those variables where you're looking a season ahead and there's just a little bit too much variability there to get a really good signal from air temperature to water temperature that then carries through to the summer. Because water temperature is a super important variable for algae productivity at short time scales, but that's typically what we found, and I think the reasoning for why that one drops out.
Okay, and you showed that if you ignore the middle predictions, that the extremes were really good for Mendota, right?
Yeah.
The high and low, and I think you suggested it was true for the broader set of lakes over the whole region.
Yeah, we do tend to get a little bit better performance in those extreme categories, and part of that is just because the normal category is often very difficult to predict, that average, because if you think about our predictors, they're either correlated with our measure of algae, going up or going down, and so some change in those predictors is going to say, oh, we think it's going to be high or it's going to be low. So it's often that we'll get kind of in that middle spot that there's the most opportunity to have a failure there.
Yeah, I just wonder if you stratified and just got rid of the middle ones and just said those don't really have a signal and only focused on the extremes, your forecast system might look a lot better and might be, show its utility a lot that way too.
That's a good point. I've not done that, but something to talk to Paul
Make
about.
yourself look good.
Yeah. Right. I was looking to do that.
Yeah, so have you tried to see if the predictors from the first part of your talk correspond to the remote sensing results from the satellites
Oh, I see.
retrospectively? Can you predict what the satellites show? Because you've got
okay.
several years of satellite data, right?
I see,
And
yeah.
you could lump the whole lake. You take the weighted average of the spatial coverage of the lake.
Yeah, no, that's a great idea. We have not done that yet. The satellite works a little bit newer too, but part of that motivation too was to be able to look back in the time series like you suggested. Those satellites were also launched in 2015 and 2016, so we don't have as much data for that, but that's a good point, something we'd definitely like to get to.
I feel compelled to say that, GeoXO, when it's launched, has an ocean color instrument on it with 390 meter resolution in the 2030s. So the follow-on to GOES are. Since you're in this building, I feel like I should be mentioning this. And I'm just curious, since it has similar resolution to Sentinel-3, but significantly better temporal resolution, it'd be interesting to see what in 10 years when you have the data.
GeoXO? you said?
GeoXO is the follow-on to the to GOES east and GOES west.
Okay, I had not known that. Thank you.
Let me ask another simple-minded question. I don't sure know anything about machine learning tools, but so if you have a relationship from a neural network or a random forest model, can you do forward prediction with that? In other words, take the correlations from the last X years between spatial variability and reflectance, and then measure reflectance the next year, and propagate that into a prediction.
Yeah. I know people have done relatively short-term prediction with that kind of thing, often based on autocorrelation. So measuring, the phycocyanin, and then next day, what do we think it's going to look like? We tried something kind of adjacent where, we're interested in these beach areas, and we were interested in seeing if these satellite passovers could capture like high phycocyanin in a beach area on a day that the public health department tested the lake and tested the beach and closed it for high microcystin. So we wanted to see if we could match those two things up. And often that was a little bit forward-looking or a little bit backward-looking because it's hard to get a satellite pass on the same day. And those did not work very well. So that's not quite, I think, the prediction piece, but we found that the spatial component really threw it off there. I mean, the temporal component really threw it off there.
Any other questions? All right, let's thank Max again.